Good afternoon, everyone. Welcome to the ESDR Kitchen Sweet and Sour episode, where two protagonists will debate a topic in the field of skin science and dermatology. This episode will take approximately half an hour and will be moderated by Professor Christina Zielinski from the Leibniz Institute in Jena and me, and I work in Leiden University Medical Center. We have a really interesting topic today. It concerns the pathogenesis of vitiligo, and more specifically, is vitiligo to be understood as a skin disease that develops as a result of dysfunction of the melanocyte, or is it primarily caused by abnormal T-cell mediated immunity? We have two outstanding speakers, leaders in their field, Julien Seneschal from Bordeaux, who will talk about vitiligo from the perspective of T-cell mediated immunity, and Mauro Picardo from Rome, who will explain the melanocyte perspective. Each of these two experts, professors, will give a 10-minute expose, and after that we'll have 10 minutes of discussion. Please submit any questions you may have for the speakers or about this topic using the chat or the Q&A function in Zoom. It's a pleasure for me to introduce the first distinguished speaker, Professor Julien Seneschal. He's a professor of dermatology at the department in the University Hospital of Bordeaux and the National Reference Center for Rare Skin Disorders. He's responsible for managing the Skin Inflammatory Disorders Unit and the Vitiligo Clinic. His research focuses on skin immunology, and he established the immunodermatology team at the University of Bordeaux with the aim of gaining a better understanding of the immune pathogenesis of vitiligo. Dear Julien, could you please share your slides?
So thank you very much, Remco. So I think you see my slide now. So thank you very much. So it's a real pleasure to be part of this ESDR webinar. So discussing about the pathomechanism of vitiligo. Of course, I will be on the side of the role of the immune response in vitiligo. So first, what are the arguments for that? Because genome-wide association studies shown that many genes associated with vitiligo are related to the immune response. So explaining that vitiligo is an autoimmune disease and explaining why we can observe in patients with vitiligo some autoimmune comorbidities or also in their family members. Second, if we think about how the immune response could be present in the skin of patients with vitiligo at early stage, mainly the goal is not to only work on what could happen in the perilegional skin of vitiligo, but also in the normal appearing skin. It's what we have done. And first, when we look at the number of CD8 positive T cells in the perilegional skin and the non-legional skin, of course, we found an increased number of CD8 positive T cells, both at the epidermal layer and in the dermal layer in the perilegional skin. But then, when we compare non-legional skin and control skin, we've already found the presence of these CD8 positive T cells at higher number compared to control skin. Then we used single-cell RNA sequencing and compare what's it's observed between perilegional skin and non-legional skin. And when we focus on the cluster of T cells isolated from perilegional skin and non-legional skin in the same patients, we observe a similar transcriptional profile between the T cells isolated from non-legional skin and perilegional skin for each patient. We also found that the cluster of T cells from the isolated from the non-legional skin and perilegional skin were also pretty similar. And by using TCR sequencing from analysis done on non-legional skin and perilegional skin, we observe actually similar T cells clone between perilesional skin and the non-lesional skin, suggesting that the non-lesional skin is probably armed with T cells as similar as we can observe in the perilesional skin. Then we focused on the CD8 positive T cells here. We identified 8 clusters of T cells, and the cell proportions was pretty similar between the non-lesional skin and the perilesional skin. And when we check for the expressions of genes of pro-inflammatory cytokines, such as important for vitiligo immune pathogenesis, such as interferon gamma and TNF-alpha, again, for each cluster, the expression of the genes of interferon gamma and TNF-alpha was pretty similar between non-lesional skin and perilesional skin. So just to say that in the non-lesional skin, we found higher CD8 T cells infiltrate in the perilesional skin compared to the non-lesional skin, but we observe similar T cells subsets, similar transcriptional profiles, similar antigen specificity, and the capacity of the cells to produce pro-inflammatory cytokines. So we can probably hypothesize that pathogenic memory T cells may already be present in the whole body skin and maybe at an early stage. And what could be the impact of this vitiligo pro-inflammatory microenvironment due to the presence of these T cells on melanocytes? And when we look, so what we did, we actually took some biopsies from perilesional skin of patients with vitiligo, and we stimulate the T cells inside the skin by using anti-CD3. And then we measure the level of pro-inflammatory cytokines in the supernatants. We found that in active patients with vitiligo, the T cells are able to produce both interferon gamma and TNF-alpha, but we found a significant level of Th2-related proteins such as IL-13. Compared to psoriasis, mainly related to interferon gamma and IL-17, IL-22, atopic dermatitis mainly associated with the production of type 2 related cytokines, or lupus mainly associated with production of interferon gamma. And then we took this vitiligo T cells, what we call the vitiligo T cell secretome, and we activate with culture that with primary culture of human keratinocytes, and we found that the keratinocytes were able to respond to this vitiligo T cell secretome by increase the expression of genes related to inflammations such as CXL9 and 10, important for the recruitment of T cells. But importantly, the melanocytes were also able to respond to this vitiligo T cell secretome by increasing the expression of pro-inflammatory genes such as, again, 6L9 and 10, but also IL-8 that has been associated with the recruitment of a subset of CD8 positive T cells. But again, when we focus on the impact of this vitiligo T cell secretome on human melanocytes, we couldn't observe that this vitiligo T cell secretome was able to induce melanocyte death, as shown here by the expression of annexin 5 on melanocytes culture with this vitiligo T cell secretome, but this vitiligo T cell secretome was able to impact melanogenesis, as shown here by the decreased expression of genes associated with melanogenesis, such as DCT, or impact melanocyte adhesion, but I will talk about that later regarding the adhesion of melanocytes. Again, here we found that the vitiligo T cell secretome impacts the expression of oxygen-reactive species on vitiligo on human melanocytes, and melanocytes culture with the vitiligo T cell secretome induce high expression production of intracellular cytoplasmic ROS, but also mitochondrial ROS, suggesting that vitiligo pro-inflammatory microenvironment is able to impact melanocytes and increase the production of ROS in melanocytes. So here, just to conclude that the vitiligo T cells located in vitiligo perilesional, but probably also non-lesional, create a pro-inflammatory microenvironment that is able to impact keratinocytes, but also melanocytes that express the receptor of these cytokines, such as interferon gamma and L13. And the melanocyte is not only a target in vitiligo, but also could respond to induce the production of chemokine ligands, important also for the recruitment of T cells. And just here is to show that one of the mechanisms associated with the loss of melanocytes is not just the melanocyte death in vitiligo, but we observe that in the perilesional skin of patients with vitiligo, melanocytes could be detached from the basal layer of the epidermis, and it was due again by the pro-inflammatory microenvironment in vitiligo, because the pro-inflammatory cytokines such as TNF-alpha and interferon gamma was able to induce the production of a protease called matrix metalloproteinase 9, mainly by keratinocytes that induce the cleavage of a protein called ikaderin, which is the main protein responsible for the attachment of melanocytes to the basal layer of the epidermis, leading to their detachment. Just to conclude about the role of this immune system, and probably the link with the talk of Mauro Picardo, is just to show that in the non-lesional skin, there is still the presence of, I didn't talk about the innate immune cells, but mainly also the presence of memory T cells that are able to create probably, that are functional and probably create in the normal appearing skin of patients with vitiligo pro-inflammatory microenvironment that can impact melanocytes. And in the perilesional skin, the infiltration of these T cells are really important and induce the loss of melanocytes. So I would like to thank you for your attention. And now I let Mauro Picardo to talk about melanocytes, the role of melanocytes. I would like to thank my vitiligo clinic in University of Bordeaux, and also of all my research group focusing the research on T cells, and also Katia Boniface, which is a co-leader of the group. Thank you very much.
Thank you, Julien.
Also, thank you very much from my side. It was very exciting to hear this very fresh new results, many of them unpublished. So this is, of course, always a big honor to be exposed to first data. Now we come to the other side of the vitiligo story, to a more melanocyte-focused talk. And it's really my great pleasure to introduce Mauro Picardo. So Mauro is a professor of dermatology in Rome at the international university called Unicamillus. And he's also a researcher at the Dermatopathological Institute Immacolata in Rome. He has been the scientific director of a dermatological institute in Rome called San Gallicano, and also the director of the cutaneous physiopathology laboratory, and the director of the Integrated Center of Metabolic Research, all in Rome. So he has really been, showing leadership in many institutes related to cutaneous research. His main topic of research are pigmentary disorders, in particular vitiligo, where he studies the underlying biochemical and also metabolic mechanisms of this disease. And maybe based on this interest, it's not so surprising that he was also chairing the first Vitiligo International Symposium in Rome, which was in 2016. I think from his CV, it also becomes very clear that Mauro is a big fan of Rome. So many things happened in Rome in his life. But he has also been, internationally very visible. And it's also an honor to mention that he has also been the president of the ESDR in the past. So without any further ado, I will hand over the stage to Mauro to give his presentation.
Thank you very much for this nice introduction. I hope to be short as Julien was. Probably I will take some a little more slides, because certainly to speak on non-immunological aspect of a disease that is defined immune-based is not necessarily so easy. And so just to check out, looking for any cartoon published about vitiligo, is considered that oxidative stress intracellularly or extracellularly could be a possible role, but without any specific definition where the oxidative stress can generate it and how this oxidative stress can have a role in the appearance of the manifestation. Just to speak about the possible pathogenesis has been reported also in the recent review by Khaled Ezzedine and journalists. They remind that basic translational and clinical studies indicate that both immune-mediated mechanism and melanocyte stress likely contribute synergistically with underlying genetic influence, and that some data suggest that there is a role of exposome in the appearance of the clinical manifestation. But just to remind that at the beginning, the capability to grow melanocyte, particularly melanocyte from vitiligo, from the year 90, it was demonstrated it was more difficult to culture this melanocyte, that there are an elevated level of cellular stress. There is the activation of the unfolded protein response. There is a 90% of reactive oxygen species. And basically what induced the capability to grow melanocyte from individual with vitiligo was the presence of catalase in the culture medium, suggesting that in any case, the generation of intracellular oxidative stress may regulate the capability of proliferation of the melanocyte of this individual, indicating that it could be some inherited defect in the situation. So Julien specified how not necessarily the immune response is capable to kill the melanocyte, but probably the mechanism could be secondary to the generation of metalloproteinases from the keratinocytes. And even if you take in consideration that also in psoriasis, we can observe some lymphocytic infiltrate with the lymphocyte primate towards some melanocyte antigen, while we do not observe in psoriasis a permanent depigmentation. If it's some transient hypopigmentation can be observed in these patients. This is probably another suggestion to say that there is some intrinsic defect in these cells that made the cell more susceptible to any indirect or direct mechanism related to the presence of immune cells. So basically, our group, since the beginning, tried to identify what happened in the pigmented skin of vitiligo patients, because the white spot is not the disease. The white spot is the scar of the disease. The disease is the disappearance of the melanocyte. So just to look what happened in the normal appearing skin could be useful to understand which could be the step of the disappearance of the melanocyte. What we demonstrate that vitiligo melanocyte produce a significantly more active oxygen species in vitro without any stimulus. They produce significant lower level of ATP. That looking for the normal appearing skin, it can be observed some sign of aging with the overexpression of P53 and some of the protein that are under the control of P53. And looking for the mitochondria, we described the mitochondria in melanocyte are widely distributed through cytoplasm and larger and spherical structure in vitiligo melanocyte, indicating the some damage on the mitochondria. And then we will confirm that just with rescued by cardiolipine alteration, we were capable to demonstrate that this melanocyte increased the generation of ATP, decreased the production of PGG1 alpha, and decreased the expression of P53, indicating that just try to improve the quality, the function of the mitochondria, some of the biological behavior of the vitiligo melanocyte can be changed. That it seems to me quite interesting situation. So also just to identify if the mechanism could be associated with melanogenesis, as sometimes has been reported in the literature, we try to evaluate if a similar effect can be observed even in cell other than melanocyte. This is a cartoon from a recently published review on fibroblasts together with the Chinese group. And just to summarize what has been demonstrated for fibroblasts taken from pigmented area of vitiligo patients. These fibroblasts present a senescent-like phenotype, including myofibroblast transdifferentiation and elevated senescence-related biomarkers, including P16, P21, and HP1. There is an increase in autophagy biomarkers, and there is a significant increase of SAPs, just to contribute to the diffusion of the aging phenomenon, as well as a significant level of metalloproteinases. And in our model, we were capable to demonstrate that the supernatant of vitiligo fibroblasts were capable to alter the expression of E-cadherin. So in our idea, can contribute to the detachment of the melanocyte and the possibility to observe in the skin, in the pigmented skin of patients with vitiligo, with some detached melanocyte in upper level of the basal situation. The fact that not necessarily exclusively melanocyte or fibroblast could be more susceptible to any the triggering factor has been confirmed by the paper from Thierry Passeron and Maurice Toulic that demonstrated that these innate lymphoid cells taken from even from the pigmented skin are more susceptible in producing cytokine, pro-inflammatory cytokine to different type of external stimuli, indicating that there is a sort of pre-activation of the pigmented skin ready to respond with an inflammatory reaction to any possible dangerous stress event. After demonstrating that there is alteration in melanocyte and fibroblast, either from the biochemical metabolic point of view and functional situation, we checked for the role of keratinocytes, that in any case is considered one of the major players to induce the disappearance of the melanocyte. What we observed that vitiligo keratinocytes present some alteration in the differentiation process, either with calcium or high level of the cells, that spontaneously, just for the stress induced by the culture, release interleukin-1 beta, 6CL10, so they are ready to produce inflammatory mediators. And we have used even culture of keratinocyte at the fourth or the fifth passage. So it seems that in any case, they maintain this alteration if this alteration has been received in vivo or if it's related to some intrinsic alteration. But we have also demonstrated that together with the alteration of the differentiation process, there is also in the pigmented skin of vitiligo patients an alteration of the constitution of the epidermal lipids. So there is an alteration of the barrier function with a significant decrease of the ceramide and a higher level of free fatty acid on the epidermal lipids, indicating as well as atopic dermatitis that there is some alteration of the barrier function. This indication of this increased sensitivity of keratinocyte from vitiligo patients has been confirmed again by another paper from Maurice Toulic and Thierry Passeron that have shown that exposure to house dust mite antigens, the vitiligo keratinocyte produce significantly more interferon gamma, TNF-alpha, and chemokines than keratinocyte for normal individual, and that the adding of ceramide that we have demonstrated are deficient in the skin may improve this type of response. So basically, in agreement, in our view, what we have described in our keratinocytes. Just to summarize, what we were capable to demonstrate in the skin of vitiligo patients that there is an increased thickness of the stratum corneum, reduced ceramide, the reduced level of antioxidants, an increased pro-apoptotic 6CL3 beta on the melanocyte, the possibility to define or to identify detached flying melanocyte, a reduced E-cadherin expression, and that there is a larger mitochondrial dysfunction, an increased rostro generation, and a stress-induced senescence phenomenon. Just to demonstrate that the oxidative stress can have a role in the progression of the disease is this paper published again by Passeron Group showing that gliadin-protected superoxide dismutases, in addition to phototherapy, was capable to significantly improve the response to the phototherapy, suggesting that in any case, reducing the oxidative stress, we can have a better recovery or a better capability of the melanoblast to differentiate in melanocyte to induce repigmentation. So what has been recently demonstrated among the different possible associations of vitiligo that metabolic or a pro-metabolic abnormalities can be detected in most of the patients with vitiligo. We have published, and several groups have published that there is an increased resistance to insulin. Frequently, there is a higher level of cholesterol, low level of vitamin D, and frequently also a high level of homocysteine, indicating it's of metabolic syndrome also in patients with vitiligo has been described in other inflammatory skin diseases. So we will try to identify if this can have a role in the metabolic aspect of the melanocyte. And what we have demonstrated that Vitiligo melanocytes have a lower response to insulin, and they have a lower level of mitochondrial DNA that have been disturbed by the expression of the presence of insulin. That is, ATP production is compromised by this insulin or IGF-1 treatment, just indicating that there is in the cell some resistance to insulin. And that if we use pioglitazone, that is a PPAR gamma activator capable to improve the glucose metabolism, we were capable to improve most of the biological properties or activities of the cell. In vitiligo melanocyte, we were capable to induce an increased production of ATP, to reduce the JC1, so the membrane depolarization, to reduce the level of reactive oxygen species, and also to reduce the marker of the senescence, and also to reduce the capability to produce inflammatory mediators from the melanocyte of vitiligo patients, indicating that in any case, the improvement of the metabolism in the cell improve the biological behavior of the cell. Then we demonstrated that also is true also for vitiligo fibroblast, that reduce the expression of SAPs after the treatment with the pioglitazone, and improving the glucose metabolism, and also they were capable to increase reduce the level of the fibronectin production. And also the keratinocytes improve in their function, improving the differentiation process, reducing the capability to release inflammatory mediators spontaneously, or also after the different stimuli, indicating that in any case, the metabolic alteration is something that seems to be inherited defect of the cells and capable to modify the biological behaviors. So in conclusion, what we can say, our studies what are suggesting that mitochondrial parent activity is strongly impact the vitiligo cell energy. Insulin growth factor 1 resistance at several levels involving dermal and epidermal cells of vitiligo patients occur. The general dysfunction of the mTOR signaling is associated with the vitiligo insulin resistance that are quite different feature independence of the microenvironment. Keratinocytes appear to be particularly susceptible to glucotoxicity and orchestrate the major process involving inflammatory the immune mechanism. And based on this study, it's possible to develop conceptually new treatment also inspired to diabetes and insulin sensitizer. So we now know that now there is a lot of attention about the immune metabolism and also the modification of the metabolism of the immune cells where they're activated and where they're resting. We suggest that in any case, our data support this type of approach and support the idea that we can improve sort of autoimmune mediated disease, just improving the metabolism rather than exclusively to inhibit the immune response. Thank you very much for your attention.
Thank you so much for sharing your insights, Professor Picardo, and also Professor Seneschal, of course. I think we will need a few more minutes than what is allotted to us to give the topic the discussion it deserves. I would want to invite the audience to post questions also at this stage after the presentations have been completed. Maybe I can start with a first general question, maybe in particular for Mauro Picardo, that is. So the treatment of the skin with monobenzone is known to elicit vitiligo, which in itself is it proves the principle that tinkering with the target cell can elicit an autoimmune disease. If we now look at the genome-wide association studies in which susceptibility genes have been identified, are there any genes that are not linked to immune cells, so genes that are expressed specifically in melanocytes that may give an insight or a clue to what the actual vulnerability in the melanocyte might constitute?
Yeah, there are some studies indicating that there are some genes that are not necessarily related to the immune response that could be altered in vitiligo patients, some polymorphism they can present. The monobenzone can produce some depigmentation, but monobenzone, a generally phenolic compound, could be also the possible source in what has been defined chemical-induced vitiligo. So it could be that in any case, the model of monobenzone should be something in support of our idea that, just disturbing the metabolism of the melanocyte in subject with our prone to develop an autoimmune response because of polymorphism could be the generation of the mechanism and the appearance of the pigmentation in even area where monobenzone was not treated.
Thank you.
Yeah, I have a question to Julien. Yeah, thank you very much again for sharing this nice data. You made this beautiful comparison of lesional and non-lesional, and as well as healthy skin, and made the point that you already see some abrogations of immunity in the non-lesional skin, and even shared CD8 T cell clones. I was just wondering how you can conclude that these non-lesional areas might be precursors of emerging vitiligo lesions. Could it also be the other way around, that what you find in non-lesional skin is maybe a spillover of the ongoing inflammation or immune reaction happening in already established vitiligo lesions? I wonder about, is it really, or have you followed up whether in the non-lesional skin a vitiligo area with depigmentation will occur to really make the point that maybe this is, a starting sign of the disease?
Yeah, so one part of your question, I probably at the end of your question, I couldn't hear because it was but anyway, I think I heard one major part of yes, because what we did, we clearly isolate the skin in the non-lesional compartment, so very far from the lesional skin, and of course, because we used patients with active disease, so you never know what's happened in the is it also due to probably the presence of also a systemic involvement of inflammation that could also participate on the lesional skin? But actually, so your question is of course great, because that means that in patients with active disease, the non-lesional compartment is not normal, and what we observe in the perilesional skin could be observed in the non-lesional skin. So, but what I did not say is that probably, so it's part of the paper we are in revisions, we have some regulatory control in the non-lesional skin that could also control probably the activation of these T cells. So even if we have similar clones, similar functionality, similar cluster of T cells in the non-lesional skin, there are some regulatory control that could dampen the activation of these T cells in the non-lesional skin. It's what we have found as mainly to explain why we do not have some lesions in the non-lesional compartment.
Mm-hmm. Okay, thank you very much.
I see within the Q&A there are a few questions by anonymous attendees. One is, I think that is for both, are there metabolic or transcriptomic differences between lesional and non-lesional melanocytes that could explain disease progression? So it follows up a bit probably on the melanocyte part of the question that Professor Zelensky just posed to Julien. Julien, do you have data on melanocyte metabolic and transcriptomic difference between lesional and non-lesional?
For me, when we actually look, because we have another data, so we did show that actually when we look at the cluster of melanocytes between the non-lesional skin and the perilesional skin, it's not easy, because in the perilesional skin, the melanocytes are not so viable. Of course, that's a problem for the analysis in the single cell, because in the quality of the single cells, you have to remove some cells with mitochondrial abnormalities, etc., etc. So we think that we lose some part of the melanocytes in the perilesional skin because they were not really viable. Because it's perilesional skin, and we know it's the target of the disease. So we found a reduced number of melanocytes in the perilesional skin, which is, of course, expected. But when we check for the differences between remaining melanocytes in the perilesional skin and melanocytes in the non-lesional skin, and we did see different clusters of melanocytes, and some show some abnormalities, but it was pretty similar between the perilesional skin and the non-lesional skin. But with the limitation that in the non-lesional skin, it's not so easy to analyze remaining melanocytes because they are targeted and probably not detected.
Right. And maybe for Professor Picardo, a related question. So the difference seems to be a bit difficult technically. But
yeah, it's difficult technically. And up to now, we have not taken in consideration to evaluate perilesional skin, just because in perilesional skin, certainly there is some influence of the inflammatory process that is around the clinical manifestation. So our aim to try to identify if there is something that could be far from a clear, evident immune infiltrate around the lesion, so to identify if there is some intrinsic situation. So we have not data from this point of view.
But a related question is that Julien Seneschal suggested that part of the melanocyte alterations are related to reactive oxygen species that are produced in the mitochondria secondary to the immune infiltrate, whereas you make the point that some of the melanocyte metabolic alterations might be pre-existent and not
this is our hypothesis
to the immune infiltrate. Is there any you may want? Is there any data to more strongly support your point of view that there is a starting kind of defect in the melanocyte instead of something that is secondary to the immune infiltrate?
Yeah, if the results of the immune infiltrate should be the generation of pro-inflammatory mediators capable to modify the biological behavior of the melanocytes, altering the capability to grow and altering, the generation of reactive oxygen species and also the expression of some membrane antigens, why the presence of this infiltrate in normal appearing skin doesn't work. Julien has explained that with the presence of some regulatory T cell. But what regulates the difference in the distribution of regulatory T cell in one lesional area toward non-lesional area, which is the mechanism of the diffusion of the regulation of that. It could be, in our idea, even just to support what happened in the Cooper phenomenon, that just a moderate or even very small trauma can induce the keratinocytes that are more susceptible to release a significant amount of pro-inflammatory cytokines and chemokines capable to cope the immune cells just in the area where the lesion appeared. And the indication that certainly in any case we are capable to improve the situation, just modify the glucose metabolism in any case, suggests that probably this type of approach can improve the type of the treatment for the disease, or can improve the diffusion of the manifestation.
Yeah, you just alluded to the question, why would T cells in non-lesional skin not lead to a lesion, but why do they do in the lesion? So what is the trigger? But maybe one step further, what is actually attracting the T cells in the first place to the lesion where the disease occurs? Is there a stimulus that is, helping T cells to cluster, become clonal and pathogenic? So what comes? So if one argues that maybe the T cells come first and then maybe they cause the damage to the melanocyte, what would be the initial stimulus attracting the T cells? And why in these very special areas, which are usually around cavities? Does microbiota play a role, or what are your ideas? Question to both, maybe.
Maybe I can start, but maybe for the second question, because there is a paper published 2 years ago now in Nature showing that there is a subset of fibroblasts that populate the skin differently. And it has been shown in many, in human skin, but also in many mouse models. And they show that there is a presence of population of fibroblasts that are located in some areas and that could respond to stimuli, pro-inflammatory or danger signal. And these fibroblasts can produce can respond also to interferon gamma, and can produce CXL9 and 10 to recruit population of T cells. And these fibroblasts populate the skin in a bilateral way, explaining why they try to explain why vitiligo is symmetrical in some areas such as elbows, knees, etc. For the first part, my thought is that in a genetically predisposed patients, mainly associated with genes associated with immune response, probably there is initial triggers targeting the keratinocytes and melanocytes that will activate, at least Mauro showed about activation of innate cells such as ILC1 that can produce interferon gamma, but also other dendritic cells that can induce as a first way migration of T cells through mainly CXL9, CXL16 or CXL10, recruiting T cells expressing CXCR3, which is the ligands of CXL9 and 10. And then there is a memory residency program, and the resident memory T cells, we know that at each time the skin is stimulated, they populate and they increase their number during the life. So my thought is that this resident memory, the pathogenic resident memory T cells are present at early stage, even before the starting of the first lesions.
Mm-hmm. Okay, thank you.
Maybe one question, if I may, and we're nearing the end of our opportunity to discuss this interesting topic, is that you've looked, or at least Julien, you've looked at the vitiligo T cell secretome, but I'd expected more something like there must be some kind of a T cell mediated cell killing that involves death receptors or interferon gamma or perforins and granzyme B. Is that something that you find? Is it kind of a classical recognition of a presented antigen and a CD8 positive T cell mediated response? Is it like that, or is it different? Is it a different type of immune response that occurs?
No, you're right, actually, because a lot of people, of course, think that actually melanocytes are killed by the immune system through the cytotoxic protein, just like granzyme B or granulosin, etc. But what we tested that actually, and we tested first, we tested the level of production of these cytolytic proteins by vitiligo T cells. But we did not show that only in vitiligo, but we compare that to other chronic inflammatory disease, mainly associated with death of the epidermal cells, such as lupus, cutaneous lupus, but also necrolysis epidermal toxicity. And we found that the T cells are able to produce this, in vitiligo, are able to produce these cytotoxic markers, but at much lower level than cutaneous lupus, T cells isolated to cutaneous lupus, or drug reactions. And then when we took this vitiligo T cell secretome compared to chronic lupus, the level of this granzyme B and granulosin was lower. And then when we culture the melanocyte plus this vitiligo T cell secretome, we couldn't find that could induce melanocyte death. So of course, we are not saying that it's not a mechanism involved in vitiligo. We say that probably there is a different way to explain the loss of melanocytes in vitiligo, and not just the killing of melanocytes.
Okay. I have a very last short question for a short answer to Mauro.
Yeah.
This is, it's a question from the audience. Is the MMP activation MMP9 specific, or are there other MMPs that are activated?
We have demonstrated mostly that is MMP9 activated.
Okay, great. And thank you very much. I think we are coming to an end now. There are a few final slides that I just want to share so that you are prepared for the next meetings. Maybe before I go to this slide that you see at the moment, I would like to announce that the next Sweet and Sour Kitchen will be on the 7th of July. most likely with the topic of immunotherapies and CAR T cells, excitingly also in autoimmune and skin diseases, so something very new. But here I would also like to announce the next upcoming meeting as part of the ESDR Kitchen series, and this will be held on the 9th of April. And the topic will be psoriatic arthritis, and the speaker, Francesco Ciccia. So we're coming to the next slides. So here you can see the whole range of different sweet and sour or different kitchen series. I would also like to mention that everything will be available online, but for this current episode, you would have to wait until Monday, and then you will also see this talk online. A final remark also to the upcoming Future Leaders Future Leaders Academy. There's still the possibility to apply until next Monday, to my information. And I would also like to point out that the next meeting, the next ESDR meeting, is coming up in September, and there's you see the important dates. And you can still apply. Maybe a final remark that there will be really great speakers in this next upcoming ESDR meeting, and you can see two of the great speakers also having been our guests. Mauro and Julien will also attend the ESDR meeting. So with this, I thank you very much again, also on behalf of my co-speaker Remco. It was a really exciting session, I think, for all of us, hopefully, and I'm looking forward to seeing you again in July. Bye-bye.
Bye-bye.
Bye. Bye. Thank you.
Thank you.